Assalamualaikum warahmatullahi wabarakatuh and good day everyone. I am Hanabala Haji and in this video I will report about endoplasmic reticulum storage disease or ER storage disease, for short ERSD. So, the endoplasmic reticulum represents the cell's quality control site for accurate folding of secretory and membrane proteins. Quality control is achieved through the association of ER chaperones with unfolded or misfolded polypeptide chains. In the ER stress response, upregulation of chaperones occurs as a consequence of misfolded proteins accumulating in the ER lumen. If these proteins fail to assume their native structure, they are retained in the ER and targeted for degradation by the proteasomes. ER storage diseases are a group of genetically based disorders in which mutant proteins fail to pass the ER quality control. Because all eukaryotic cells contain the ER, the clinical phenotype of ERSD is very heterogeneous. Disease may result from the mere lack caused indirectly by toxic effects of the misfolded proteins or aggregates thereof in the cell. Additionally, the cell's reaction to the ER stress may include signaling pathways which are ultimately detrimental. Experimentally, ERSD serves as models to study the cellular reactions to a variety of perturbations. In particular, understanding the links between ER stress and cell degeneration may give valuable insights into the pathogenesis of other diseases where the accumulation of indigestible toxic material leads to cell injury. Storage diseases, defects in degradation or in biosynthesis. So, storage diseases are caused by the intracellular accumulation of indigenous compounds which directly or indirectly impair cellular functions and may even lead to cell death. A well-known group of these disorders includes the lysosomal storage diseases, which are caused by the accumulation of molecules from the cell exterior in lysosomes. And another one is this endoplasmic reticulum, ER storage diseases. In contrast, ERSD are characterized by the accumulation of proteins in the very first compartment of their normal biography because of defects early in biogenesis. Synthesis of all but a few mitochondrial proteins is initiated at cytosolic ribosomes. Proteins normally functional in the ER or the Golgi apparatus at the plasma membrane or in the endosomal lysosomal system, or which are destined for secretion from the cell, are first targeted to the ER, the major site of insertion proteins into the lipid bilayer and of translocation across the membrane. So let's take a look at this figure. Figure 1 illustrates the major routes of membrane transport originating from the ER in eukaryotic cell. Correctly folded proteins exit in transport vesicles which bud from the ER and congregate at the ER Golgi intermediate compartment. ER Golgi intermediate compartment, ERGIC, by vesicles or tubules, they are delivered to the C side of the Golgi apparatus and move by vesicular transport or cisternal maturation through the Golgi cisternae to the trans-Golgi network, to the TGN, trans-Golgi network. From here, different pathways diverge. Number 1. Constitutive secretory vesicles to the cell surface. Number 2. Secretory granules of the regulated secretory pathway in endocrine or exocrine cells. Number 3. vesicles to endosomes containing mannose-6-phosphate receptors and their cargo, lysosomal enzymes that have been tagged by a mannose-6-phosphate modification in glycans. In contrast, proteins are able to acquire their native conformation accumulate in the ER, at least to some extent. In most cases, after some lag period, they are degraded by a process called ER-associated degradation. This is what we call ERAD for short. ER-associated degradation, ERAD. The machinery for ERAD is located in the cytosol. Functional defects could occur at any of the transport and sorting steps described above. What we described above. For example, mutation in the signal sequence affecting membrane translocation or signal processing may cause familial hypoparathyroidism, coagulation factor X deficiency, Crigler-Najjar syndrome type 2, familial central diabetes insipidus, or chronic pancreatitis. Only few instances of impaired protein exit from the Golgi apparatus are known, and the term Golgi storage disease is not commonly used. One example is autosomal dominant nephrogenic diabetes insipidus. In adult autosomal dominant polycystic kidney disease, the polarized traffic of proteins from the transgolgi network to the basolateral membrane in proximal tubular cells is impeded. This sorting and transport defect appears to be caused by a deranged cytoarchitecture resulting from complex interactions of proteins involved in cell-cell adhesions with mutated proteins named polycystins. The vast majority of mutations, however, affect protein folding in the ER. ER retention may occur in any cell type, explaining the wide variety of the clinical spectrum of the ERSD. In this review, we will associate disorders focusing on some clinically relevant examples. So, in the plasmic reticulum, there's activity na nangyayari. The protein translocation and folding, ER exists versus the ER-associated degradation, ER stress response, the unfolded protein response, and ER overload response, and ER stress and its link to programmed cell death. So, let's proceed to the selected examples of ER storage disease. Ito yung mga examples ng mga diseases ng ER storage. ERSD can be divided into several pathogenetic groups. In the vast majority, the defect lies within the cargo, such as the secretory protein encoded by a mutated gene. So, itong table na to, makikita natin dyan yung different example ng ER storage disease. So, isa na dito, one of these is the cystic fibrosis. So, cystic fibrosis in short for CF. Cystic fibrosis is an autosomal recessive disorder affecting the pancreas, the exocrine and endocrine insufficiencies, bronchial glands, chronic lung infections, and emphysema. And also the biliary cirrhosis, and this it's also affecting the intestinal glands, meconium ileus, and also the reproductive organs and sweat glands. So in our era, lung disease clearly determines the course and life expectancy in cystic fibrosis patients. The CF gene was identified in 1988 by positional cloning. It includes large A168 kDa, epithelial membrane protein called cystic fibrosis transmembrane conductance regulator. It's so very mahaba, right? So for short, CFTR, cystic fibrosis transmembrane conductance regulator, which functions as the chloride channel and regulator of other ion channels. So, yung lalaki na merong CF are infertile because of the absence of function vasa differentia. Conversely, heterogeneous CFTR mutations can cause an isolated infertility syndrome called congenital bilateral aplasia of the vas deferens. These patients present without the other phenotypic hallmarks of CF. A number of studies in heterologous expressions. A number of studies in heterologous expression system have documented the CFTR DF508 and other mutations associated with CF fail to mature and are trapped in the ER or ERG. In culture cells of various type, both wild type and yung sinabi natin na DF508 is associated with calnexin and other chaperones, but only the wild type molecule can escape from this association and traffic through the Golgi to the plasma membrane. In contrast, itong DF508 CFTR na fails to exit the ER and after retrotranslocation to the cytosol is degraded by the ubiquitin proteasome system. The defect in protein maturation and ER exit appears to be temperature sensitive. If itong DF508 CFTR is expressed in cells grown at low temperatures, 30 degrees Celsius or below, a proportion of molecules escape and are transported to the plasma membrane. The same effect can be obtained by strongly overexpressing the mutant protein or by treatment of cells with so-called chemical chaperones such as glycerol. Interestingly, patch clamp studies have demonstrated that this DF508 CFTR localized in the ER or plasma membrane retains its function as chloride channel. This confirmed the notion that the CF phenotype associated with DF508 CFTR appears to originate from mislocation and premature degradation, rather than altered function properties or decreased stability of the mutant CFTR. A1, alpha antitrypsin deficiency. So, it is an acute phase protein synthesized by the liver. It is the most abundant serine protease inhibitor in plasma. The clinical pictures of A1 deficiency varies with zygosity and the type of mutation present. Heterozygous individuals, yung heterozygous na tao may be asymptomatic carriers but can have a predisposition to obstruct lung disease at later ages. In patients homozygous for a null mutation, no A1PI will be produced or secreted into the bloodstream. These individuals are at risk for bronchiectasis and pulmonary emphysema due to the unopposed proteolytic activity of neutrophil elastase in the lung, which kung saan naglilid ito ng chronic tissue and destruction. Certain other mutations lead to a change of function of the A1PI molecule. For example, due to the replacement of arginine for methionine replacement of arginine for methionine at position 358 na yung APY natin is yung APY natin ay loses its antitrypsin activity but obtains heparin independent antithrombin activity, causing the phenotype of a severe bleeding disorder. Finally, so-called deficiency mutations direct the synthesis of an aberrantly processed molecule. The most important member of this class is the allelic variant PIZ, replacement of the glutamic acid by lysine at position 342, which is the most frequent A1PI mutation overall with an allele frequency of 0.01 to 0.02 in Caucasians. Plasma levels of this disease, A1PIZ homozygous patients are greatly reduced but measurable. Homozygotes are prone to develop early pulmonary emphysema, and in a subset of patients, 15%, cirrhosis and hepatoma. It was shown nearly 30 years ago that hepatocytes in patients with A1PI deficiency contain large insoluble intracellular inclusions. It is now known that the majority of PIZ proteins, itong protein na PIZ proteins ay approximately 85% are retained in the endoplasmic reticulum in patients and in transgenic mice expressing the human PI gene. On the molecular level, the reactive centrilobe, the base of which harbors the amino acid substitutions, folds into a sheet of a neighboring proteins, that allowing so-called lobe sheet polymerization and leading to the formation of large aggregates. Several papers have reported that the degradation of this ER-retained material occurs through the proteasomes in a ubiquitination-dependent manner, although other data suggested that in hepatocyte degradation pathways other than the proteasomes may be involved. Mutants other than A1PIZ have also been found to be ER-retained by co-precipitation with the ER chaperones calnexin or immunofluorescence and immunoprecipitation experiments. So another disease also is the congenital goiter and hypothyroidism due to the thyroglobulin deficiency, or in short for TG, is a large homodimeric glycoprotein and forms the major protein constituent in follicular luminal colloid, where it serves as storage pool for thyroid hormones. Upon the THS stimulation, colloid is taken up into the follicular cell by endocytosis. Subsequently, itong thyroglobulin is hydrolyzed in phagolysosomes in leaf thyroid hormone molecules are released into the bloodstream. TG is synthesized in the thyrocytes and secreted into the follicular lumen via the regulated secretory pathway in a TSH-dependent manner. The ER chaperones, BPI, GRP94, and calnexin have all been shown to interact with TG during the normal folding process. In humans, ito yung mga humans na mayroong autosomal recessive TG deficiency is one of the causes for congenital hypothyroidism and goiter. The molecular pathogenesis of this disorder has been studied in an animal model, the so-called COG over COG mouse. In thyrocytes cultural ex vivo from overcog glands, mutant TG is not secreted in the culture medium but it is retained in the ER, where kung saan yung levels of 5 different chaperones are elevated 5 to 10-fold compared to thyrocytes isolated from wild-type controlled mice. Yung the cause for this ER stress response is the failure of mutant TG to fold properly and form homodimers. Likewise, in humans with congenital TG deficiency, TG is retained in the ER. Thyroid colloid is largely devoid of TG, and the ER is distended when studied by electron microscopy. Thus, unlike in A1 antitrypsin deficiency, unlike siya sa A1 antitrypsin deficiency with liver disease or familial neurohypophyseal diabetes insipidus, the retained TG in these instances of congenital hypothyroidism does not seem to cause cellular damage. Rather, the phenotype is explained by the mere absence of TG and hence circulating thyroid hormone. So now let's proceed here in the diabetes insipidus. Autosomal dominant hypophyseal diabetes insipidus, ADNDI. This is a rare disorder caused by defects, single or multiple base deletions, missense and nonsense mutations in the gene encoding the antidiuretic hormone. So, mababasa natin dito sa PPT yan. So, mababasa natin yan dyan. Nasabi dyan is, ayan. So, other example of ERSD is diabetes insipidus inherited nephrogenic diabetes insipidus. So, nephrogenetic diabetes insipidus, results from resistance to the action of AVP at the concentrating segment of the nephron. It is most commonly acquired but may also occur as an inherited disorder. So another one is the autosomal recessive protein C deficiency. Thrombotic disorder, protein C Nagoya is an allelic variant which fails to exceed the ER. And the compound heterozygosity for two mutations in the pro-hormone convertase 1 gene, which includes an endopeptidase, was found in a female suffering from extreme childhood obesity and showing elevated plasma levels of various pro-hormones which were not produced due to the lack of enzyme. The mutation caused the protein to be retained in the ER. And another disease then is the tyrosinase deficient albinism. Mutation in tyrosinase cause enzyme misfolding, prolonged association with the ER chaperones calnexin and calreticulin, and ER retention and in melanocytes. And another one is the osteogenesis imperfecta. It is a group of bone matrix disorders due to inborn errors of metabolisms of collagen synthesis. In fibroblast cultures obtained from a deceased infant, ER transset of the mutant protein was retarded and it was partially degraded by ERAD. Interestingly, assembly and secretion into extracellular matrix of collagen triple helix containing mutant strands occurred, but collagen content of the matrix was greatly reduced. And that's all for this report. Thank you.